thought I'd just make a quick video to summarize the process that you've gone through with the online lab. So if you haven't already, work your way through the lab first and then watch this video back later just to check your understanding and make sure that you completely understand the steps that you worked through and what you were setting out to achieve in this practical. Okay, so first of all, the overall aim of this prac is gene cloning. What the scientists would be setting out at the start of this experiment would be to clone a gene. The gene that we are interested in cloning in this prac was something called ANT-R. What was the phenotype of the ANT-R gene? That is the bacteria that possesses the ANT-R gene becomes resistant to antibiotics. And it's a gene that I've referred to before, and it's usually a gene that is a selective gene. It's not usually the gene of interest. It is usually one that's used in the selection process for bacteria that have taken up our gene of interest. But in this case, it is the gene of interest. So that's our first difference between this prac and what I've previously spoken about. Okay, the steps that they worked through in order to clone this gene. You are familiar now with the processes that were involved along the way. Okay, so step one, bacteria that were resistant to antibiotics already, they were isolated. And it was found that there was a gene located on one of their plasmids that was incurring somehow their resistance to the antibiotic. So this is the gene that allows a bacteria to be antibiotic resistant. The scientists want to clone this gene. To do that, the gene needs to be isolated. So it is isolated, it is cut out of the plasmid. That ANT-R gene gets cut from the plasmid and it undergoes PCR. So you would have seen in the interactive the graph demonstrating the steps of PCR, and you're already familiar with those. And at the end of the PCR process, we've amplified the DNA, and we've now got lots of copies of the gene to use. But we want more. We want more copies, and so we can do that through our more natural approach to gene cloning. Okay, so we've got lots of copies from PCR. The next step then is to get our gene into a lab plasmid, a plasmid that we can use for our bioengineering purposes. Okay, so to get our antibiotic resistance gene into a lab plasmid, we need to use a restriction enzyme to cut open the plasmid. And that restriction enzyme needed to be the same one that was used to cut the gene from its natural plasmid. Why? Why does it need to be the same restriction enzyme? So that the sticky ends match and it can ligate together to form our recombinant plasmid. Okay. You then did this step. So hang on, I'll just flick across one. Step 4 is to conduct the gel electrophoresis and talk about why that's important in a second. But the steps that you went through were to load the plasmid, the gene, and the hopefully recombinant plasmid into our gel chamber, and then run the electrophoresis to see our banding pattern that would result. And so if I go back to the results that you then saw, these were the results that came up in your little interactive there. So in our gel, we had the standard, the gene by itself, the ANT-R gene, and the plasmid that we were using, which had been linearized, and the hopefully recombinant plasmid. So this step is important because the recombination process is a bit hit and miss. Sometimes the plasmids form the recombinant plasmid, and sometimes these sticky ends simply ligate back together, and then we don't have the gene inserted. So what we're doing here in step 4 is checking to see if our plasmid has taken up the gene, if it's truly a recombinant plasmid. So what evidence do we see of that in our gel electrophoresis? Number one, the recombinant plasmid is one band. If this mixture had resulted in the plasmid rejoining and the separate gene, we would see a smaller band representing the gene and a bigger band representing the plasmid, but they would be two separate bands still. So the fact that we see just one band here is evidence that our plasmid has formed a recombinant plasmid. And not only that, when you've done your measurements, you have mathematically proven that the size of the gene and the size of the plasmid, when you add them together, it comes up to be the same size as this band here, which is our recombinant plasmid. So we've done that to double check that we have formed a recombinant plasmid at this stage. So our gene has successfully been inserted and ligated into our plasmid. Okay, you then moved on to step 5. So step 5 was our transformation process. So taking our recombinant plasmid there now, mixing it with our bacterial culture and shocking it, undergoing a process of electric shock or heat shock. When we've mixed them together, we're hoping now that we've got some transformed bacteria that have taken up our recombinant plasmid and now have that gene for antibiotic resistance. The test that we've done for that you've done for that in the interactive, was spreading the bacteria onto agar plates. Now, there was 2 different agar plates that we use. One has no antibiotic on it, and the other has antibiotic present on it. So when we spread our bacteria onto these 2 plates, think about this plate for a second. If it's got no antibiotic, it doesn't matter if the bacteria has our gene or not, it will grow. So this is being used as a control here just to show what the bacterial growth should look like if the gene is really playing no part. So with no antibiotic present, all of our bacteria should be able to grow. And you have to look kind of closely at it. The very cloudy surface of this agar plate means that we've got what's called a lawn growth of bacteria. There's no distinct colonies because there's just so much bacteria growing on this plate. So this is not no bacteria, this is covered in bacteria. So this is a lawn growth of bacteria. Whereas the one with antibiotic present, only the bacteria that have successfully transformed and now have our antibiotic resistance gene, they will be the only ones that are capable of growing on this agar plate. And so we see distinct colonies. Distinct colonies have formed on the plate with antibiotic. So these are all our bacteria that have our gene of interest. And the other representation you have of this test is showing a streak pattern across a gradient agar plate. So we can see bacteria that are antibiotic resistant, that they have the gene. They will grow not only in the no antibiotic side of the agar plate, they are able to grow into the higher concentration where we find antibiotics. The ones that do not have the antibiotic resistance gene, is that streak ends, so it's only able to grow where there's no antibiotics, and as soon as we start getting a trace of antibiotics in the agar plate, their growth ceases. Okay, so what was the overall point of this experiment? It was to clone the antibiotic resistance gene. And you achieved that through, Melbourne University achieved that through first conducting PCR, creating a recombinant plasmid, checking that recombinant plasmid was successful with gel electrophoresis, and then performing a bacterial transformation to then clone lots of copies. Every bacteria that's present on this agar plate would have a copy of the antibiotic resistance gene. So successful gene cloning technique.